Dear students, today we will talk about histology of the organs of endocrine system. Endocrine system is one of the regulatory systems which provide regulation of all vital functions of our organism. Two basic systems which provide regulation is nervous system and endocrine system, and also immune system is described as one of the regulatory systems. It provides immune supervision. And here we can see two basic types of regulation. It's nervous regulation and endocrine regulation or humoral regulation. They have some similarities and some differences. So let's look and let's revise nervous regulation. There are nervous cells or neurons. They generate nerve impulses and they run on their processes, axons, and they achieve target cells. So we have regulatory cell and target cells, they receive direct impulse from neuron, and this connection is provided by processes of nerve cells. So nerve cells, they have very long processes which achieve target cells and send them some impulses directly. There are neurotransmitters and they are released via synapses and there are synaptic contacts, and it gives information to target cells and they react to those impulses. And here we can see synapses. It's axon, nerve ending of nerve cell. It's postsynaptic membrane of target cell. It may be neuron or another cell, for example, secretory cell or muscular cell. And here we can see nerve impulse. It goes on the cell membrane. It comes here to the nerve ending. And here we have vesicles of the neurotransmitter. There are molecules with neurotransmitters, special substances which are produced in the nervous cell. And after this, those vesicles, they release their content to the synaptic cleft, and molecules of neuromediators, they are attached to the receptors which are located on the postsynaptic membrane. They activate them and it causes some changes in the target cell. For example, if this cell is nervous cell, it causes a generation of new wave of depolarization. It's a transmission of nerve impulse. And also we have endocrine regulation or humoral regulation. In this type of regulation, we also have cells which provide regulation, and also we have target cells. And in both cases, in nervous regulation and in endocrine regulation, this distance between regulating cells and target cell may be very big. And in this case, in nervous regulation, we have direct contact between neuron and target cells. But in endocrine regulation, we haven't direct contact between regulatory cell and target cell. And how do they provide their regulation, those endocrine cells? They produce some substances which are released in the bloodstream. Those substances are known as hormones, and bloodstream carries hormones to the target cells, and target cells, they receive this information from endocrine cells and they react to those information, and it causes some changes in the metabolism or some functions of the target cells. So here we can see endocrine regulatory cells. They produce some substances. They are hormones. We have very big number of different hormones, and they provide specific regulation of different functions. And those hormones are produced by the cell, and cell releases them via exocytosis, and those hormones are released directly in the bloodstream, and blood carries those hormones and they achieve target cell, which may be very far from secretory cells. And here we can see target cell and hormones, they come here and they achieve target cell. And this regulation is specific because target cells, they have receptors, and receptors, they react only to specific kind of hormone. That's why endocrine regulation, humoral regulation is very specific. And those hormones, they achieve numerous different cells, but they have impact only on target cells. Only target cells may react to those hormones because they have receptors. And this attachment of hormones respond to receptors causes some changes in this cell, and usually it's changes of metabolism of cell. Usually it's increase of metabolism, but it may cause decrease of metabolism. It may activate mitotic divisions of the cell, and cell will begin mitotic division, or even it may activate cell suicide. Cell will activate own apoptosis and will die. So there are different effects which are caused by different hormones, and it's general plan of endocrine regulation. So we have two basic kinds how do target cells receive those impulses, information, and now we will talk about endocrine organs, endocrine cells, and how do they provide regulation and how do they produce different substances. So usually endocrine cells, they are secretory cells, and more endocrine cells, they have epithelial origin. They derive from epithelial cells, from epithelial tissue. And here we can see covering epithelium, and some epithelial cells, they are exocrine or endocrine, they have synthetic activity. All cells of our body, they have ability to produce some substances, but only secretory cells, they are specialized in that, and they have well-developed rough endoplasmic reticulum, smooth endoplasmic reticulum, and also they have well-developed granules which contain secretory product. And here we can see covering epithelium, and it may contain usual covering cells of epithelium, and some cells may be secretory. Here we can see basal membrane, which is present in all kinds of epithelia, and here we can see capillary, which is located near the epithelium. And here we can see blood flow, and blood carries different substances, different amino acids, sugars, and other substances which are necessary for the metabolism of the cells. And for example, here we can see substances, they may be amino acids which are necessary for the synthesis in the cell. For example, this cell is secretory cell, and it uptakes those amino acids, and they are going to rough endoplasmic reticulum, where they are used to produce proteins. And those secretory molecules, they are going through transport vesicles to Golgi apparatus, which provides processing, final processing of synthesis. And then Golgi apparatus forms secretory vesicles, which contain different secretory products, for example, polypeptides. In this case, here we can see them. And secretory cells, they have vesicles or granules with secretory product, and they release their content via exocytosis. It's secretory cycle of the cell. This cell is exocrine or exocrine cell. It releases secretory products on the surface of epithelium, on the surface of our body, or in the lumen of different cavities, for example, luminal organs. And exocrine cells, they release the secretory products usually in outer environment. It's exocrine secretion. And we also have another type of secretion which is known as endocrine. Let's look how this type of secretion is provided. So here we can see different amino acids also, for example, and they are coming to the cell. Cell uptakes them and uses them for the synthesis of also, for example, polypeptides, proteins. Rough endoplasmic reticulum provides this. Then those molecules are going to transport vesicles, and they bring those secretory products to Golgi apparatus, which provides processing of those substances, and also forms secretory vesicles which contain final proteins or in other substances which are known as hormones, secretory products of endocrine cells. And let's look at appearance of these cells. Exocrine cells, they usually have secretory granules or secretory vesicles near their apical pole. And endocrine cells, they have granules near the basal pole because they release their content via basal pole of the cell. And they release their secretory products directly in the bloodstream. And here we can see how do they release hormones, and they are going to bloodstream, and bloodstream carries those hormones to the target cells. So here we can see two types of secretory cells, exocrine and endocrine cells, and some of exocrine and endocrine cells are located directly in the covering and lining epithelia, in epithelial tissue. And here we can see exocrine cell and endocrine cell, they are located in the covering epithelium, and those single endocrine cells, they are known as single hormone producing cells. Those endocrine cells which produce hormones from amino acids are known as upward system because of abbreviation of amine precursors uptake and decarboxylation. And also there are single steroid producing cells. But we also have endocrine organs which are composed of endocrine cells. Not only single cells are present in our body, we also have endocrine organs. And endocrine organs, they are glands. So let's look how endocrine glands are formed and what is the difference with exocrine glands, and let's look at the structure of them. So here we can see covering or lining epithelium, which is composed of usual epithelial cells, and in this place we can see formation of invagination tubule which is growing in the underlying connective tissue, and this tubule goes deeper, and some cells of this tubule may become to be secretory cells. They produce something, and it's formation of a simple tubular gland, exocrine gland, because it has duct. This duct provides excretion of those secretory products. This secretory unit is tubular in this case, and it may be acinar. When cells change their appearance, they are bigger and they form round structures. It's typical structure of small exocrine gland. It has secretory unit and it has excretory duct. And here we can see capillaries, nerves which provide regulation and innervation. And what is the difference between exocrine and endocrine gland? Here we can see endocrine gland. So what is different? First of all, duct disappears. And those glands, they haven't ducts, they are ductless, they have only secretory units, and they release their secretory products directly in the bloodstream. So they have very close contact with capillaries. They are located near endocrine cells, and also endocrine cells, they have granules which are located near their basal pole, and they release their content via basal pole in the bloodstream. So here we can see typical structure of secretory unit of endocrine gland. And here we can see secretory units of the gland, and in exocrine gland also we have ducts, branched system of ducts, and there are epithelial structures which form secretory units, and also ducts. They are epithelial structures and they are specific in all exocrine glands. And this epithelial specific part of the organ which provides functions of the organ we call parenchyma. It's main specialized part of the organ, parenchyma. And also we have stroma. Stroma is composed of loose and dense fibrous connective tissue which fills the spaces between secretory units, and it maintains them, it provides nutrition, protection, and here we can see loose fibrous connective tissue which surrounds groups of secretory units, and it forms lobules of the gland. Here we can see lobulated structure of the gland. And also between the lobules we have thin layers of dense connective tissue, and also dense connective tissue forms capsule which covers entire organ, and together dense and loose fibrous connective tissue, they form stroma of the organ. So we have parenchyma specific part and non-specific part stroma, which is similar in all exocrine and endocrine glands. So here we can see typical structure of exocrine glands. And what will be different in endocrine gland? Endocrine glands, they are ductless, so ducts are absent. we have only secretory units, and they release their products in the bloodstream, but also endocrine glands. They have parenchyma, which is formed by secretory units, and also it has stroma, loose fibrous connective tissue inside the lobules, and septa, connective tissue septa, which separate neighboring lobules, and also capsule is present here. So it's a typical structure of endocrine gland. And we have two basic types of secretory units in endocrine system, in endocrine glands. It's follicle and trabecule. Here we can see follicle. It has cavity. It's bubble-like structure. It's vesicular structure, and it has cavity which is filled with something, with secretory product, but it's ductless. And also there are trabecules. They are also composed of endocrine cells. Also, they haven't duct, but they haven't cavity. They are rows of cells, cores of cells, but they haven't cavity, and we call them trabecules. And different glands, they may have follicles or trabecules. Usually thyroid gland, it has follicles, and all others, they have trabecules. They are structural and functional units or secretory units of those glands. And here we can see structure of thyroid gland lobules, which contain follicles, secretory units, and typical structure of gland with trabecular structure, with trabecular organization. Here we can see trabecules, and they are surrounded with loose fibrous connective tissue. There are septa and capsule. It's a trabecular structure of endocrine gland. Endocrine organs, endocrine glands, they have different functions, they have different localization, and there are different classifications, and we will discuss this classification. So, first of all, we divide endocrine organs into two big groups. We have central organs, which regulate functions of some peripheral organs: hypothalamus, hypophysis, and epiphysis or pineal body. They have close relationships with nervous system. They are literally part of the brain, and they provide communication between nervous system and endocrine system, and also they regulate some organs, peripheral endocrine organs. And also we have non-central endocrine organs, which include clearly endocrine organs. They are called peripheral endocrine organs: thyroid gland, parathyroid glands, and adrenal glands. They aren't central organs, they don't regulate in other organs, but they have only endocrine function. They provide only synthesis of hormones: thyroid gland, parathyroid glands, and adrenal glands. They are peripheral endocrine organs. Also, we have organs with endocrine and non-endocrine functions, and also they may be described as a kind of peripheral endocrine organs. They are pancreas, thymus, gonads, testes, ovaries, kidneys, and placenta. They have two or more functions. One of them is endocrine function. Another function may be exocrine. For example, in pancreas, it produces pancreatic juice. It's exocrine, non-endocrine function, and also it provides synthesis of some hormones. Thymus, it's organ of immune system. It provides differentiation of T lymphocytes. It is involved in the functions of immune system, but also it has endocrine function. Gonads, testes, and ovaries, they provide synthesis, they provide formation of spermatozoa and ova, but also they provide synthesis of male and female hormones. Kidneys, they provide excretory function. They are organs of the urinary system, but also they have endocrine function. And placenta, it provides communication between embryo and mother. It provides nutrition of embryo, but also it has ability to produce some hormones. And we'll discuss those organs in the following topics when we will study their non-endocrine function. Today we will focus on clearly endocrine organs, peripheral and central. And also we have single hormone producing cells. They are of two kinds: steroid producing cells and acute system. Acute, this abbreviation means amine precursors uptake and decarboxylation. So cells of immune system, they use amino acids for the synthesis of their hormones. Both kinds of single hormone producing cells, they are distributed in different organs, usually in epithelia, covering and lining, but sometimes they are present in other places. We'll discuss them when we'll study different organs, and we'll study which cells have endocrine function. So today we'll focus on central endocrine organs, which are regulating organs. They regulate functions of the peripheral organs, and completely clearly endocrine organs, which are thyroid gland, parathyroid glands, and adrenal glands. And here we can see peripheral, completely endocrine organs, thyroid gland, parathyroid glands, and adrenal glands. And we'll begin from the smallest glands. They are parathyroid glands. Usually we have 4 glands, 2 right and 2 left, lower and upper, but some people they have 2 glands, right, 1 right and 1 left. Here we can see localization of parathyroid glands. They have connective tissue capsule. Sometimes this connective tissue capsule is common with thyroid gland, so common capsule covers both glands. Structural functional unit of parathyroid gland, it's trabecule, row of cells which are called parathyrocytes. Here we can see row of parathyrocytes, and they haven't cavity between them. They release their secretory products directly in the capillaries which are located near them. And also we have connective tissue, loose fibrous connective tissue, which fills the spaces between trabecules. And here we can see organization of parathyroid gland. It's parenchyma, which is composed of trabecules, and stroma, which includes interlobular connective tissue, loose fibrous connective tissue, septa between the lobules, and capsule. It's organization of parathyroid glands, and we can conclude that it's the most typical structure of the endocrine glands. Here we can see trabecule of endocrinocytes. They are parathyrocytes, capillary and blood cells. And here we can find 2 kinds of parathyrocytes, this kind and this kind. So more numerous cells are called chief cells. They have basophilic staining, and they produce main hormone of parathyroid gland. It's parathormone. Or parathyrin. It's produced by chief parathyrocytes. And also we can find some cells which have oxyphilic staining, and they are called oxyphilic parathyrocytes. They have oxyphilic staining, they have pink light cytoplasm, and sometimes they are a little bigger than chief cells, and they aren't so numerous. And some scientists think that oxyphilic parathyrocytes are old chief cells which are located between chief cells, and they change their appearance and they reduce ability to produce parathormone. Here we can see organization of parathyrocytes, branched structure of trabecules. They form rows and they have multiform shape. And here we can see parathyrocytes, chief parathyrocytes, and also oxyphilic parathyrocytes here, and capillaries between them are present. It's capsule and septa, connective tissue septa between lobules. And here we can see appearance of parathyroid gland under microscope. Rows of cells which are known as trabecules, and also small, almost invisible capillaries between them. And here we can see rows of cells, and here we can find follicles of thyroid gland, because they have very close localization, and we cannot even find border between 2 glands. What is the function of parathyroid gland? Parathyroid gland chief cells, they produce parathormone, and this hormone, it regulates calcium level in blood. It increases calcium level in blood. And target cells for this hormone are osteoclasts of bone. they undergo this impulse, this information, they are activated and they begin resorption of bone. And they dissolve matrix of bone and calcium ions are released in bloodstream and it increases calcium level in blood. So osteoclasts are target cells of parathormone. And now we are going to thyroid gland, which is also completely endocrine gland, peripheral endocrine gland. So here we can see anatomy of thyroid gland. It has two lobes, right and left. It has isthmus and pyramidal lobe, additional pyramidal lobe. Structural and functional unit of thyroid gland, it's a follicle, bubble-like structure or spheric structure, vesicle, which is composed of epithelial cells. It's simple cuboidal epithelium, and also we have cavity inside. It's filled with special fluid, which is called colloid. And also we have basement membrane here, which surrounds this follicle because it's epithelial structure. And here we can see close contact between capillaries and endocrine cells. Capillaries, they form network which surrounds follicles. And here we can see epithelial wall of the follicle. There are thyrocytes, main cells of the thyroid gland. They form single layer of epithelium. It's simple cuboidal epithelium. It's basement membrane. Inner content of the follicle is filled with colloid, jelly-like fluid, and there are some progenitors of hormones, and later we'll discuss this. Sometimes colloid contains resorption vesicles, and also we will discuss what is the source of their origin. And near the follicles there are capillaries, usually fenestrated capillaries, and thyrocytes, they release their hormones in the capillaries. And there are neighboring follicles and loose fibrous connective tissue between the follicles. It's typical structure of thyroid gland. And here we can see lobules, which contain follicles, loose fibrous connective tissue, and septa between the lobules and connective tissue capsule of the gland. And here we can see drawing of histological slide of thyroid gland follicles with simple cuboidal epithelium, cavities of the follicles filled with colloid. And also we can see here groups of cells which haven't cavity. They are called interfollicular islets. Here we can see them. They are located between the follicles. That's why they are called interfollicular. So what are those islets? They may be parts of the follicles, but in another plane of section. For example, if we will cut this follicle in this plane, we'll see follicular cells without cavity. But also interfollicular islets may contain extrafollicular cells. They also are hormone-producing cells, and also stem cells are present here in the interfollicular islets. And here we can see structure, appearance of the thyroid gland, follicles, interfollicular islets between them, and epithelium of the follicle and colloid inside the follicle. And let's compare parathyroid gland and thyroid gland. Here we can see structural and functional unit of thyroid gland. It's follicle and it has cavity filled with colloid. And parathyroid gland, it hasn't cavity, which is filled with colloid. It has trabecules. Let's look at the synthesis of the thyroid hormones and how they are produced. So you know that there are thyrocytes, main cells of thyroid gland, and they provide synthesis of main hormones of thyroid gland. And here we can see thyrocytes. They produce iodinated hormones of thyroid gland, main hormones. And also we have C cells. Here we can see this C cell or extrafollicular cell. It doesn't achieve cavity of the follicle, it doesn't achieve colloid, and it produces non-iodinated hormone. It's calcitonin. So, hormones of thyroid gland are iodinated, which contain ions of iod, and non-iodinated hormones. Iodinated hormones are thyroxine or tetraiodthyronine or Tis4, and 3-iod-threonine, T3. They contain different number of ions of iod, 4 or 3, and it gives their names, T4 or T3. They have similarities in their functions and synthesis. They regulate carbohydrate, lipid, and protein metabolisms, so they activate all kinds of metabolism. They increase temperature, body temperature, they increase arterial pressure, they increase heart rate, so they activate activity of our organism, and they regulate vital functions of our organism. And iodinated hormones, they are produced by thyrocytes, typical main cells of thyroid gland. Here we can see thyrocytes. Also there are non-iodinated hormones. It's calcitonin. It regulates low blood calcium level. Here we can see cell which produces calcitonin, non-iodinated hormone, and it regulates blood calcium level. It makes it lower. And opposite hormone with opposite action, it's parathyroid of parathyroid glands. So parathyroid hormone increases calcium level, and non-iodinated calcitonin decreases calcium level. It's non-iodinated hormone. It's polypeptide which hasn't specific features of the synthesis. But iodinated hormones are produced by thyrocytes, and they have some specific features of their synthesis. So let's look at the synthesis of T3 and T4. So here we can see follicle of thyroid gland, and there are thyrocytes which produce T3 and T4. T3 and T4 are produced from amino acids, and those amino acids, it's tyrosine. It's necessary for the synthesis of T3 and T4, and also iod ions are required. And here we can see all necessary materials, and blood carries amino acids and iod ions to the thyrocytes. They uptake them from blood from the basal pole, via basal pole, and they use them for the synthesis of T3 and T4. Cells, thyrocytes, they form polypeptide chain from amino acids tyrosine. So they use only one kind of amino acids, and they produce polypeptide. You know that polypeptides proteins, they may be formed by 20 different kinds of amino acids, and thyroglobulin, this polypeptide, is produced, it's formed only from amino acids tyrosine. So here we can see this polypeptide, and this thyroglobulin, after the synthesis, is released in the cavity of the follicle. The cell produces thyroglobulin and releases it in the cavity of the follicle. And here thyroglobulin is accumulated together with water, and it forms colloid inside the follicle. So colloid is solution of water and thyroglobulin, and it's accumulated here. And next stage is iodination of oxidation of iod. Iod is oxidated, and it's attached to the thyroglobulin. It's iodination of thyroglobulin. And here we can see iodination. And let's look how many iod ions are attached to each tyrosine, to each amino acid. We can see that some molecules, they attach 2 ions of iod, and some attach only 1. So here we can see this feature. And next stage, it's uptake of iodinated thyroglobulin. When cell receives signal that it should release T3 and T4, it uptakes this thyroglobulin from colloid, and empty places which were containing thyroglobulin are called resorption vesicles. They were places where thyroglobulin was present, but thyrocytes, they uptake thyroglobulin, and empty places are visible as white round structures. And after that, here we can see appearance of resorption vesicles, and when thyroid gland is very active, number of those vesicles increases. And after that, thyroglobulin is broken into amino acids. Next stage of synthesis of T3 and T4 is hydrolysis. And thyrocyte, it cuts those polypeptide into pairs of amino acids, and we can see that some pairs, they have 2 and 2 ions of iod. It's 4 ions of iod, and it's T4. And in some cases, one amino acid has 2 ions of iod and another has only 1, and we call this T3. T3 and T4, 2 kinds of thyroid hormones, iodinated thyroid hormones. And T1 and T2 also may be produced, but they haven't biological activity. So they are produced, but may be produced, but main hormones, active hormones, are T3 and T4. And after that, they are released in the bloodstream, and blood carries those hormones to their target cells. And here we can see follicles, and after the resorption of thyroglobulin, T3 and T4 are produced, and they are released in the capillaries, which are located near the follicular cells. And here we can see T1 and T2, they haven't biological activity. T3 and T4, they are active, and they have impact in the metabolism. And also let's look at appearance of this follicle. It's typical normal follicle, and here we can see that cells, they are higher, they are enlarged in volume, and number of resorption vesicles is increased. So cells are columnar, not cuboidal, columnar, and numerous resorption vesicles. It shows us hyperfunction or increased function of thyroid gland. Here we can see increase in the function of thyroid gland. And here we can see hyperfunction of thyroid gland and appearance. Columnar cells, columnar thyrocytes, and numerous vesicles in colloid. Opposite condition, it's hypofunction. Thyrocytes are flattened in shape because they are inactive, they don't uptake thyroglobulin, they don't uptake colloid. That's why they are flattened in shape, and they may be even squamous. And also we cannot find here resorption vesicles in the colloid, and follicle is enlarged in volume because it's stretched. Cells, they accumulate thyroglobulin, but they don't receive signals that they should release their content, that they should release T3 and T4, or they may not have enough iod ions, and they cannot produce T3 and T4 from thyroglobulin. That's why this follicle is stretched. It contains a lot of accumulated thyroglobulin, but new hormones are almost absent. And it's hypofunction, appearance of thyroid gland in hypofunction. And here we can see histological slide, this hypofunction of thyroid gland with flattened thyrocytes and stretched follicle. And here we can see even broken colloid in the follicles, because in hyperfunction it becomes to be very dense because it is accumulated in storage during very long time. And here we can see thyrocytes, they are typical cells, and also we have here C cells which produce calcitonin, which regulates calcium level. And now we are going to another gland of endocrine system. It's adrenal gland. We have 2 adrenal glands, and adrenal glands, they have 2 basic parts. It's cortex, outer layer, and medulla, and they are covered with thin connective tissue capsule. Here we can see capsule of adrenal gland, its cortex and medulla. Cortex has structure of typical endocrine gland. It's formed by epithelial tissue, and it forms trabecula. Trabecula are a little bit modified. But medulla is composed of modified sympathetic neurons, and it's composed of modified nervous tissue, and it's not epithelial tissue. So medulla has bigger relationships with nervous system than with endocrine system. And we'll look at the structure of medulla. So we'll begin from the cortex. So cortex and medulla are two basic parts of adrenal gland, and cortex, it has three zones which are revealed with hematoxylin and eosin staining. The outermost layer is zona glomerulosa, and cells of adrenal gland, they form round structures which are called glomeruli. Next, zona fasciculata, there are fascicles of the endocrine cells or modified trabecula, and that's why the zona is called zona fasciculata, and the deepest one is zona reticularis. Trabecules, they form reticular system, and that's why this zone is called zona reticularis. And the deepest part which underlies cortex, it's medulla. It's located in the central part of adrenal gland. Here we can see zona glomerulosa, it's zona fasciculata and reticularis, and medulla. Zona fasciculata is composed of trabecula. They have more typical structure. Between them we have capillaries which divide rows of the cells, and they uptake hormones. Cortex produces steroid hormones which have lipid origin, and they are released in the capillaries. Here we can see structure of zona fasciculata, rows of cells or trabecula of cells, and between them we have capillaries. They are usually sinusoidal. Here we can see zona fasciculata, and the deepest zone is zona reticularis. It has reticular appearance, network-like appearance. And different zones of adrenal cortex, they produce different hormones. All cortical hormones of adrenal gland, they are steroid hormones, but they have some features. Zona glomerulosa, it produces mineralocorticoids. Main mineralocorticoid, it's hormone aldosterone, which regulates metabolism of sodium and potassium. It has target cells in the kidneys, and it activates sodium and potassium pump in kidneys. So zona glomerulosa produces aldosterone, which regulates sodium potassium pump and sodium potassium metabolism in kidneys. Zona fasciculata, the biggest zone, it produces steroids which are called glucocorticoids. They regulate carbohydrate metabolism. They regulate metabolism of glucose and in other carbohydrates. They increase sugar level in blood, and they may increase arterial pressure, and also they decrease allergic reactions. They decrease hyperactive immune reactions. So there are effects of glucocorticoids. And zona reticularis produces sex hormones, female and male, and you know that there are testes and ovaries which main function, one of the main functions, it's production of female and male hormones. But also those hormones are produced in adrenal glands. They are only glands which produce those hormones during puberty, and also they are main glands which produce hormones in old age, but also they produce sex hormones during adulthood. And a feature of zona reticularis is that female and male hormones are produced in both in females and males, but in different amount, in different proportions. So zona reticularis also is additional zone, is additional organ which produces sex hormones and in female and male organisms. And also you can find here fourth zone, it's sudanophobic zone, which is located between zona glomerulosa and zona fasciculata. This zone, it's called sudanophobic zone because this zone may be revealed using Sudan staining. Sudan staining stains, it reveals lipid inclusions inside the cells. Hormones of adrenal cortex, they are lipids, they are steroids, they have lipid origin. So they are stained, they are revealed with Sudan. But if we apply Sudan staining, we can see that zona glomerulosa has granules, zona fasciculata and zona reticularis, they have granules which are stained with Sudan. But this zone, one zone, thin zone between zona glomerulosa and fasciculata, hasn't granules, and this zone isn't stained with Sudan, and that's why it's called sudanophobic zone. Why cells don't have granules, they don't have steroids? Because they are stem cells and they don't produce hormones and they provide regeneration of another zones. So it's a regeneration zone which is composed of stem cells, and this zone is revealed only if we apply special staining, for example Sudan, which reveals inclusions of steroids in the hormone producing cells. And under the cortex we have medulla. Medulla is composed of modified neurons. They are modified postganglionic sympathetic neurons, and they produce noradrenaline or norepinephrine and adrenaline or epinephrine into the blood. Together, both kinds of cells, they are called chromaffin cells. They react with chromatosols. Noradrenaline and adrenaline, they have a lot of similarities in their biological role, and I think you can say a lot about effects of adrenaline. So when you feel fear or when you are excited, you can feel all effects of adrenaline. And noradrenaline, it has almost the same effect. So it's excitement of the nervous system, it's increased arterial pressure, increased heart rate, and they activate organism, they activate nervous system and sympathetic part of the autonomic nervous system. So there are effects of adrenaline and noradrenaline which are produced and released in the adrenal medulla. And here we can see adrenal cortex and adrenal medulla, there are two basic types of adrenal gland. Cortex is composed of epithelial tissue and medulla contains modified neurons. So we have discussed peripheral endocrine organs, thyroid gland, parathyroid glands, and adrenal glands, and now we are going to central organs, central endocrine organs, which regulate functions of some peripheral endocrine organs. They are hypothalamus, hypophysis, and epiphysis or pineal body. And we'll begin from hypothalamus and pituitary gland or hypophysis. They are located here, it's hypothalamus and hypophysis, and they have very close anatomical relationships and also functional relationships. Here we can see that hypothalamus is part of the diencephalon of the brainstem. Here we can see thalamus and hypothalamus, and its pituitary gland or hypophysis. And hypothalamus, it has structure which is typical for the nervous system, and at the same time hypothalamus is part of nervous and endocrine systems. It has nuclei, so nervous cells or modified nervous cells are present in hypothalamus, and they form nuclei, groups of perikaryones. And those groups, they are located in three different parts. So there are three groups of nuclei: anterior, intermediate, and posterior. Three groups of nuclei, which and nuclei are typical for the structure, histological structure of the brainstem. And here we can see anterior and intermediate nuclei, but we cannot see here posterior nuclei. It's because of feature of posterior nuclei that posterior nuclei, they are part of nervous system only. They provide only nervous functions. And anterior and intermediate nuclei, they belong to endocrine system. So they are involved in endocrine system, and we'll discuss only anterior and intermediate nuclei, which have endocrine function. And here we can see anterior, intermediate, posterior nuclei. Posterior are only nerve nuclei, and anterior, intermediate nuclei, they are involved in the endocrine system. And also hypothalamus has very close relationships with hypophysis. Here we can see hypophysis, and hypophysis, it has two principal parts. It's neurohypophysis, which is composed of nervous tissue, as well as hypothalamus. Here I can see hypothalamus and neurohypophysis, and they are composed of nervous tissue. It has pale pink shade in this picture. And here we can see another part of hypophysis is adenohypophysis. It has bright, colorful staining, different cells with different shades. It's adenohypophysis, which is composed of epithelial tissue, epithelial secretory cells. So two different parts of hypophysis and two different tissues and different functions as well. So pituitary gland or hypophysis consists of two parts. It's neurohypophysis or pars posterior. It's made up of nervous tissue, and adenohypophysis is composed of epithelial tissue, and it has three parts. It's pars distalis or anterior lobe, pars tuberalis or tuberal lobe, and pars intermedia or intermediate lobe of hypophysis. And here we can see four lobes of hypophysis. Three lobes are parts of adenohypophysis: pars distalis or anterior lobe, pars intermedia or intermediate lobe, and pars tuberalis or tuberal lobe. And neurohypophysis, it's pars posterior or posterior lobe. So four lobes and two different parts are present in adenohypophysis and neurohypophysis. So in pituitary gland as a whole. Hypothalamus and different parts of hypophysis, they have close relationships, and you should know that anterior hypothalamus has very close relations with neurohypophysis, with posterior hypophysis. Anterior hypothalamus and posterior hypophysis. Here we can see direct connection between those parts, anterior hypothalamus and posterior hypophysis. It's one system. And another system and another pair, it's intermediate hypothalamus and adenohypophysis or anterior hypophysis. Another pair of system. So anterior hypothalamus, posterior hypophysis, and intermediate hypothalamus and anterior hypophysis or adenohypophysis. And now we'll discuss those pairs and we'll look what are those relationships. So, neurohypophysis and anterior nuclei of hypothalamus are the first pair, and we'll discuss them first. So, neurohypophysis and anterior nuclei of hypothalamus. Here we can see anterior nuclei of hypothalamus, and they are composed of atypical nervous cells. They are called neurosecretory cells. They are neurons with perikaryon, and they produce some substances, some hormones, not neurotransmitters. There are hormones, vasopressin or antidiuretic hormone and oxytocin, which are produced by those cells. And those cells, they have very long axons which are going to neurohypophysis. Here you can see neuron, its cell body, and long axon which goes through stalk of hypophysis and achieve neurohypophysis. So neurosecretory cells. And they don't produce neurotransmitters, they produce hormones. Hormones are going through axon and they achieve axon ending. And here we can see that axon ending doesn't send information to another cell. They have contact with capillaries, and this is called exovasal synapses. And let's look here. It's axon ending. It's like axon ending of the nervous cell. It's capillary. And here we can see inclusions of hormones which are produced in neurosecretory cells. And after transporting of those secretory products of those hormones to the axon ending, they are released in the bloodstream. It's exovasal synapse. It's specific feature of hypothalamus, of neurosecretory cells of hypothalamus. They have intermediate position between nervous cells and endocrine cells, typical endocrine cells. And after that, hormones are carried to the target cells. And here we can see neurosecretory cell action potential goes here, and it's typical nerve action and atypical in the neurosecretory cells when hormones are released in bloodstream and blood distributes those hormones and they achieve target cells. And before axon terminal, there are wide regions of the axons where inclusions of hormones are accumulated. Those regions are known as herring bodies. Sometimes even axon terminals and exovasal synapses are described as herring bodies. So entire ending of axon may be described as herring body. And here we can see herring bodies in the pars nevrosa or posterior hypophysis. It's neurohypophysis. And also there are pituitary glial cells which are present here. Neurohypophysis, it doesn't produce own hormones. Hormones of anterior hypothalamus are released here. Own hormones in neurohypophysis are absent. It's only place of release of vasopressin and oxytocin into the bloodstream. And what are the effects of vasopressin and oxytocin in our organism? Vasopressin, or antidiuretic hormone, it's hormone which regulates water balance and it keeps water in our organism. It has target cells in kidneys and it decreases release of water, so it activates reabsorption of water and amount of water in our organism is increased. And another effect is vasoconstriction. Cells of the blood vessels, they are sensitive for the vasopressin and they undergo contraction and it causes an increase of arterial pressure. And another hormone which has the same way of release, it's oxytocin. Oxytocin is known as hormone of female reproductive system because target cells of oxytocin are present in uterus and in other organs of female reproductive system. There are smooth myocytes which are contracted under influence of oxytocin, and it plays very important role during delivery when uterus is contracted. And also there are myoepithelial cells in mammary gland and they are contracted under influence of oxytocin and it helps in lactation. But males, they also have cells which are sensitive for oxytocin, and there are smooth myocytes of male reproductive system. For example, smooth myocytes of the prostate gland. They are sensitive for oxytocin. Now let's go to another pair of the hypothalamus and hypophysis parts. It's adenohypophysis or anterior hypophysis and intermediate nuclei of hypothalamus. You know that adenohypophysis has pars distalis, intermediate, and tuberalis, and those lobes, they have close relations with intermediate hypothalamus. So let's begin from adenohypophysis, and let's look at this structure. You know that neurohypophysis doesn't produce own hormones, it only releases hormones which are produced in the anterior hypothalamus. It's composed of nervous tissue. But adenohypophysis, it has another structure. It's composed of epithelial tissue and it's composed of epithelial cells which produce different hormones. They produce own hormones. And here we can see trabecules, which are composed of adenocytes of adenohypophysis. So cells of adenohypophysis are called adenocytes. And here we can see structural and functional unit of adenohypophysis. It's trabecule of adenohypophysis. There are capillaries, blood cells, and another trabecules. And we can see here that we can find three different types of cells. Some cells are pale, they are very light, they aren't well stained. And some cells are well stained, some of them are pink or red, and some of them are blue or violet. So we have three types of cells, and those cells which are pale, which we call chromophobes. Pink or red cells we call chromophils, oxyphils, and those blue cells we call basophils. And oxyphils and basophils together, they are called chromophils because they are well stained. And cells which aren't well stained are called chromophobes. And here we can see chromophobes. They are pale, they are the lightest cells. Chromophils, acidophils, and chromophils, basophils. Why do they have those staining And what is the feature? Because those cells, they have granules which accumulate hormones before they are released. They are granules, and they are going to release those granules in the bloodstream. And different hormones, they have different staining. So if hormones have oxyphilic staining, they are stained in red or pink colors. We call those glands, those cells, oxyphils or eosinophils. And if cells have granules with basophilic staining, we call them basophils. And some cells, they don't have granules with hormones. Some of them are stem cells, inactive cells, or some of them are chromophils which have released their granules and at the moment they are empty. So here we can see chromophobes, here we can see pale cells, chromophils, and oxyphilic chromophils, and basophilic chromophils. So here we can see classification of adenohypophysis cells, chromophobe cells and chromophil cells. They have granules, they are active, and chromophobe cells, they haven't granules, they are non-active. And chromophil cells are divided into two groups, acidophil cells and basophil cells, according to their staining. But acidophil and basophil cells, they have own kinds. So acidophil cells, they include two kinds, and basophil cells, they have three kinds of cells. Acidophil cells are somatotrophs and mammotrophs. Basophil cells are thyrotrophs, gonadotrophs, and corticotrophs. So main in their names, it's trophs. There are troph cells. And first part of name means which hormone is produced. So somatotrophs, they produce growth hormone or somatotrophin or somatotrophic hormone. Mammotrophs or lactotrophs, they produce prolactin or lactotrophic hormone. Thyrotrophs, they produce thyroid stimulating hormone TSH or thyrotrophin. Gonadotrophs, they produce follicle stimulating hormone FSH and luteinizing hormone LH. And corticotrophs or adrenocorticotrophs, they produce adrenocorticotrophic hormone ACTH or corticotrophin. And those hormones, they may be troph hormones. They may be called troph hormones because almost all or more troph hormones, they have target cells in the peripheral endocrine organs. For example, thyrotrophs, they have target cells in thyroid gland. Thyrocytes, they are sensitive for TSH. Also in other endocrine cells of the follicles of the ovaries and also cells of the testes, they are sensitive for FSH and LH, so they are under control of gonadotrophs. Cells of zona fasciculata and zona reticularis, mainly zona fasciculata, they are under control of ACTH, and zona reticularis, it's under control of gonadotrophs of FSH and LH. So different peripheral endocrine organs, they are controlled by troph hormones. So here we can see basophils and acidophils or oxyphils, and also there are chromophobes, they are pale. And here we can see numerous acidophils. We can find region where acidophils are very numerous, and they are of two types. First type, it's somatotrophs. They produce growth hormone, and target cells for growth hormone are present in bones, in cartilage, in connective tissue, and it increases growth of our organism, mainly during childhood, somatotrophic hormone. And another hormone which is produced by another oxyphilic cells, it's prolactin or lactotrophic hormone, which activates lactation in mammary gland, production of milk in mammary gland. Basophil cells are thyrotrophs, gonadotrophs, and corticotrophs, and here we can see basophil cells. So basophil cells, thyrotrophs, they produce thyroid-stimulating hormone TSH or thyrotrophin, which activates thyrocytes in thyroid gland and activates synthesis of T3 and T4. Gonadotrophs, they produce FSH and LH, and their target cells are in the ovaries and in the testes. And in topics female and male reproductive systems, we'll discuss features of those cells, target cells, and how do they interact with FSH and LH. So FSH and LH in female organisms, they regulate menstrual ovarian cycle, and also they have very important role in male reproductive system. And corticotrophs or adrenocorticotrophs, they produce ACTH or adrenocorticotrophic hormone, which activates synthesis of glucocorticoids in zona fasciculata of adrenal cortex. That's why this hormone is called adrenocorticotrophic hormone. And they regulate functions of adrenal cortex. So we have here basophil cells of 3 types. Those cells, troph cells, somatotrophs, lactotrophs, thyrotrophs, gonadotrophs, and adrenocorticotrophs, they regulate some target cells in the peripheral endocrine system, some in other target cells, but also they are under control of another substances. They are under control of intermediate hypothalamus. So we have the highest level of endocrine regulation, it's intermediate or middle hypothalamus. This intermediate hypothalamus also has neurosecretory cells, which are related in their functions and structure to neurosecretory cells of anterior hypothalamus. They also have perikaryones, cell bodies, and long axons, which form exovasal synapses. But they aren't going to the neurohypophysis, they are going to the medial eminence, special region which is located on the anterior hypothalamus, its medial eminence. And axons of neurosecretory cells of intermediate hypothalamus, they are coming here. And let's look at blood supply of this region. Here we can see artery, which brings arterial blood to the medial eminence. And here we can see primary capillary network in the medial eminence. It's the place where neurosecretory cells release their hormones in the medial eminence bloodstream. Then this medial eminence keeps all those, it takes this network takes all those hormones and brings them to the portal vein of the hypophysis. Here you can see portal vein. And special feature of this region that it forms secondary capillary network. Usually we have only one artery, one vein, and capillaries between them. But here you can see artery, vein, and another vein, and 2 capillary networks, primary and secondary. Secondary capillary network is located in the adenohypophysis, and portal vein collects blood from the medial eminence and carries this blood to the anterior hypophysis, to adenohypophysis. It's needed to provide contact of the factors, regulatory factors of intermediate hypothalamus. They are released here and they are coming through portal vein of hypophysis to the target cells, and those target cells are oxyphils and basophils of adenohypophysis, adenocytes of adenohypophysis, those troph cells. They receive own regulation from intermediate hypothalamus. It releases those hormones in the medial eminence, and then portal vein of hypophysis brings those factors to the anterior hypophysis, and adenocytes of adenohypophysis, they uptake different hormones and increase or decrease own synthetic activity. And those hormones which are produced by intermediate hypothalamus are called releasing factors, liberins and statins. Releasing factors because they increase or decrease, activate or inactivate release of troph hormones by adenocytes. So their target cells are adenocytes of adenohypophysis. And there are 2 kinds of releasing factors. Liberins, they increase activity of troph cells of adenohypophysis, and statins, which decrease their activity. they are released in bloodstream in the medial eminence, and then they get to adenohypophysis. So here we can see medial eminence, and then hormones are going to the adenohypophysis. There is typical synapses, which is characteristic for the nervous system, but in the medial eminence we have exovasal synapses, which are like exovasal synapses in the neurohypophysis. We have here axon terminal, and hormones releasing factors are released in the bloodstream of the primary capillary network of the medial eminence, and then blood carries those releasing factors to the target cells, which are located in the adenohypophysis. There are adenocytes, oxyphils, and basophils of adenohypophysis. And here we can see how neurosecretory cell works. It releases hormones directly in the blood vessel. So we have two systems of the interaction, anterior hypothalamus and posterior hypophysis, and intermediate hypothalamus and anterior hypophysis or adenohypophysis. And we have different levels of regulation of the endocrine organs. So intermediate hypothalamus produces liberins and statins, and they have target cells in the adenohypophysis. Liberins activate release of troph hormones. Statins, they inhibit release of troph hormones. When liberins are coming here, they are specific, and each troph cell has own liberin, own statin. Almost all they have own liberins and statins. For example, thyrotrophs, they receive liberins, thyroliberins, and thyrostatins. Also, there are gonadoliberins, gonadostatins, somatoliberin, somatostatin. And those liberins and statins, they increase or decrease release of troph hormones. So some specific liberin comes to the adenohypophysis and activates release of some specific troph hormone. It's second level of regulation. And then troph hormones are going to the adenohypophysis dependent endocrine glands, which react to the functions, to the action of troph hormone. And then they produce and release own hormones, hormones of the peripheral endocrine glands, and they are going to the bloodstream and achieve target organs. For example, thyroid regulation. Thyroliberin and thyrostatin are produced by intermediate hypothalamus. Thyroliberin goes to the adenohypophysis and activates thyrotrophs, and they produce TSH, thyroid stimulating hormone. TSH goes to the thyroid gland, and TSH activates thyrocytes. They produce T3 and T4, and they are going to target organs. It's last level of the regulation. And these levels of regulation are called hypothalamo-hypophyseal system of regulation. Some organs are dependent on those systems, and some are independent. So we have adenohypophysis dependent endocrine glands and adenohypophysis independent endocrine glands. So they are dependent endocrine glands are thyrocytes of the thyroid gland. They are under control of TSH. But in thyroid gland we have also C cells which produce calcitonin, which decreases calcium level, and those cells aren't under control of adenohypophysis, as well as parathyroid glands which increase calcium level. So calcium level regulation isn't controlled by adenohypophysis. Adenohypophysis dependent endocrine glands and zones are zona fasciculata and zona reticularis of the adrenal cortex. Zona fasciculata is under control of ACTH, and zona reticularis is controlled by FSH and LH. But zona glomerularis and adrenal medulla aren't controlled by adenohypophysis. Gonads are under control of FSH and LH, as well as zona reticularis of adrenal cortex. Both they produce sex hormones, and also endocrinocytes of the islets of Langerhans, of pancreas, islets of Langerhans, and also single hormone producing cells in the Pute system. They are adenohypophysis independent endocrine glands. So we have these levels of regulation, and they regulate functions of some endocrine glands. And first and second levels are provided by central endocrine organs. Third level is provided by some peripheral endocrine glands. So we have discussed interactions between adenohypophysis and intermediate hypothalamus, anterior hypothalamus, and neurohypophysis, and this system is called hypothalamo-hypophyseal system of regulation. And now we are going to the last organ, central organ of endocrine system. It's pineal body or epiphysis. It's located here. It's also part of the brain. And epiphysis provides regulation of different cyclic processes in our organism. There are cells, main producing cells, they are pinealocytes. Here we can see pinealocytes. And also there are surrounding or supporting cells, astrocytes, glial cells, which help pinealocytes to provide their functions. So there are two basic types of the cells, pinealocytes and interstitial cells or glial cells, astrocytes. Epiphysis or pineal body controls all cyclic processes in human organism. Two main hormones of epiphysis are serotonin and melatonin. Serotonin is produced during day when we have bright lighting, and melatonin is produced in the darkness during night, and cyclic production of them regulate different cyclic processes. During aging, cells produce different substances, and sometimes there are accumulations of different crystals inside the epiphysis. They are known as brain sand. There are crystals inside epiphysis. It's normal condition which may occur during aging. So we have discussed structure of all organs of endocrine system, clearly endocrine organs, central and peripheral, and in other organs which have endocrine and non-endocrine function we will discuss in the following topics. So students, that's all, and thank you for your attention.